Ladies and gentlemen, Assalamualaikum warahmatullahi wabarakatuh. A very good morning and a warm welcome to the second day of the third Bioinformatics and Biodiversity Conference BBC X the International Symposium on Bioinformatics InSyB in conjunction with the second Synthetic Biology and Biotechnology SBB. We hope that our program yesterday was really beneficial to you. Please capture the moments from the conference and post them in your social media by tagging us using hashtag BBC_InSyB_SBB 2022. This conference is brought to you by Masyarakat Bioinformatika dan Biodiversitas Indonesia MABI, Persatuan Ahli Biologi Sintetik Indonesia PAPSI, Synbio ID, and Akademisi, in collaboration with Asia Pacific Bioinformatics Network AP Bionet. The event is sponsored by PT Pandu Bioscience and Klinik Karir. The aim are to facilitate the exchange of knowledge, skills, and updates on bioinformatics, biodiversity, biotechnology, and related topics among academics, government institutions, industries, and students. This year, BBC is held in conjunction with the 6th International Symposium on Bioinformatics InSyB and the 2nd Synthetic Biology and Biotechnology SBB. It's an honor for me to welcome our keynote speakers. Profesor Dr. Trina Ekawati Talei from Department of Biology, Samratulangi University, Indonesia, Associate Professor Dr. Bimo A. Tejo from the Department of Chemistry, University Putra, Malaysia, Dr. Tias Hikmawan as Head Tribe of Biotechnology, DTO, Ministry of Health, Republic of Indonesia, Profesor Dr. Insinyur Miftahuddin MSI from Department of Biology IPB University Indonesia, Dr. Daniel Christoph Volk as Senior Researcher from Technical University of Denmark. And also welcome all participants of the third Bioinformatics and Biodiversity Conference BBC X the International Symposium on Bioinformatics InSyB in conjunction with the second Synthetic Biology and Biotechnology SBB. Ladies and gentlemen, the conference will start with keynote talks from Professor Dr. Trina Ekawati Talei and Associate Professor Dr. Bimo A. Tejo, followed by keynote talks from Dr. Tias Hikmawan. And after the break, we have keynote talks from Professor Dr. Insinyur Miftahuddin MSI and Dr. Daniel Christoph Volk, followed by closing statements from BBC X InSyB 2022 Chair representing SBB 2022. The last agenda would be National Congress of Masyarakat Bioinformatika dan Biodiversitas Indonesia MABI. And if you haven't registered for the Congress, please do so at https://bbc-insyb.academisi.co.id. Remind to all participants to fill the attendance list on the link available in chat box. Distinguished ladies and gentlemen, without further ado, I now present Dr. Muhammad Yusuf to lead the first keynote talk session. Dr. Muhammad Yusuf is a lecturer and a valuable researcher for the concern of molecular modeling. He completed his doctoral program in pharmaceutical sciences from University Science Malaysia in 2015. He recently published research according to all atom molecular dynamics simulation, QSAR analysis, and pharmacophore modeling. Dr. Muhammad Yusuf, the screen is yours.
Okay. Thank you, Ibu Risa. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Very good morning, everyone. It's nice to see you all. And here, present with us, our honorable speaker, Prof. Trina Talei. It's nice to see you, Prof. Trina. So, without further delay, I would like to introduce Prof. Trina. Okay. Yeah. So this is the short resume of Prof. Trina. So Prof. Trina Ekawati Tale from Universitas of Samratulangi, Indonesia. So she is a full professor at the Department of Biology from Faculty of Mathematics and Natural Sciences in Samratulangi University. So Prof. Tale has over 50 scientific papers on projects either presented or published. She is internationally recognized in her field in conservation biology, and she's also an editor at SAFE Network, which is Asia Pacific Network for Sustainable Agriculture, Food and Energy. And she's also received a Millennium Development Goal Award in 2013 in Indonesia. So we would like to welcome Prof. Trina Tale to deliver her talk for today. So please, Prof. Trina, the screen is yours.
Okay, I hope you can hear my voice clearly.
Yeah, a little bit louder, please, Prof.
Okay, what about this?
Yeah, it's okay.
Okay, it's okay, yeah. So, good morning, everybody. Greetings from Bandung. Actually, I'm from Manado, but currently I'm in Bandung.
Oh, okay.
Yeah. It's a pleasure to be here, to be invited as the keynote speaker to deliver my presentation about the economics for the precision medicine. So I hope we all can learn about this topic that I will share. I hope you don't mind if I now share my slide. So, do you see my slide now?
Yes, clear. Okay.
Okay, then. Okay, now I will share information about the economics for precision medicine. And why is that so? Because just our treatment today is based on actually from William Osleria, and the approach is just imagine that when we go to the physician or a doctor, and then the doctor will see the clinical presentation from ourselves, and then they will do the examination, and then they will find pathological signs, and then combine these two, and the doctor will define our disease. So this was established in 19th century, that the current classification of disease by linking the principal organ system in which symptoms and signs manifest with anatomic and histopathology findings. So it is known as Oslerian paradigm. However, there are some shortcomings of this approach, because sometimes it is, when we go to the doctor to see the doctor, there is a delay in the onset of the clinical signs. Sometimes when we go to the doctor, we don't see the clear sign of what happened to our body. So this approach also doesn't usually take into account the molecular and environmental factors that actually influence the disease. So the current setting is, the doctor interpret the complaint that we share with the doctor, and then they perform a physical exam from which differential diagnosis can be determined. Then they will ask about what happened, the sign of our body, and then they will do also the laboratory examination, and then after that they will confirm what happens, what kind of disease we have. But shortcomings of this approach, it hasn't taken into account that actually the functional status of our body is driven by the expression of 35,000 more or less genes, and then also 6 million protein variants that the manifestation of this the conclusion is only informed from a few laboratory studies. So that's why the diagnosis and patient response to treatment are uncertain. Just for example, when we go to doctor, when we complain, we talk about the, we are trembling, and also we have the fast heartbeat, and we feel dizzy, and then sometimes this manifestation camouflage, between maybe anxiety and also with the heart disease. Then that's why now we are moving to the more precision medicine, because you see, we are here. We are here. We are a human race. We have very diverse races in the entire world, and then we have different genetic makeup. So our genes, and then combined with our environment and then lifestyle, it defines us. Okay, so it's not merely our genes that will determine us, our health, our appearance, but we have to consider also the environment and lifestyle. So the genes that we have from our parents, and then environment where we live, our lifestyle, all influence our health, and also our disposition to disease, and also the symptoms. And then also depend on, it will influence how well the treatment may work. For example, when a patient A have a cancer, and then also the patient B also has cancer, and they receive the same treatment. One can be cured, and the other cannot be cured. Why is it so? Yeah, because everybody has different genetic makeup. That's why this has to be also considered, and it has to be taken into account. So because our human body is a very complex system. Yeah, beside of the influence of our lifestyle, also when we see our body is can be referred as a complex adaptive system, and it can be defined as the collection of individual agents with freedom to act in ways that are not always totally predictable. So what happened in our body, the system cannot always be totally predicted, and then the action of all elements in our body, they are all interconnected. So one aspect in the body, the change in one aspect in the body, it will totally also change the context for other aspect in our body. So it is a very, very complex system, and interconnected, interrelated each other. So now we go back to the traditional medicine. Just I mentioned previously that usually we all receive the treatment for basically one manifestation of a disease. For example, a cancer patient with colon cancer receive the same therapy even though they have different biomarkers. So I will talk about the biomarkers later on. So we see patient A, patient B, patient C with the different genetic makeup, and will receive the same treatment. That's in turn, the patient A maybe have good effect on the body, and then one patient has no effect at all, and then the other patient will have adverse effects. So this treatment is very general. So the prescription of the generic medication may not be effective for all patients. So we have see all that's very obvious in usually a patient of the cancer. Yeah, I just recently lost my three colleagues this year because of the cancer, and then with different kind of cancer, but I think they all received the very similar treatments. But anyway, so now we are moving to the precision medicine, which, for example, the colon cancer patient, the same patient that we talked about previously, they will receive finally a targeted therapy based on their biomarkers. So this therapy will be based on the omics analysis for each individual, and then the patient of each patient will be examined and analyzed, and then the result will come. Each patient will will have different biomarker, and then based on this biomarker, they will receive individual treatment. So it is hoped by having diagnosed with even though with the same kind of disease, but after having information about individual biomarkers, and then they will receive different kind of treatment that is tailored for each patient, and then hopefully they will have a positive effect, and then this biomarker based on the omics information, and also environment and the lifestyle, and then also with the clinical data, they will receive most effective treatment. So in the hope that it will be significantly have the very best treatment for every patient. So this now I propose this I'm proposing this concept actually. So our system is when all of us has a genome, and then there are some variation in the genome, particularly SNP when we have a SNPs, and also not only SNP, and when during the expression of our gene, and it will be influenced also by our epigenetic tags, and then these epigenetic tags will be surely influenced by our lifestyle, and then also environment, and also the microbiome that we have, and then it will influence the expression of our gene, and then we will have the many RNA transcript, and then it will be translated into some of our RNA will be translated into proteome, and then also it will metabolize into many kind of metabolome. And this all information also together with the clinical data, and then it will be integrated into one particular data for each individual, and then based on this each individual will get a different kind of treatment based on their biomarkers. So they will maybe this also can be used as prevention, precision prevention, but also can be used as a precision treatment for each individuals. So the precision medicine strategies combine all of clinical data with omics data also, including the microbiome, and then also the lifestyle and environment to identify the precision treatment option for individuals with a given disease. So this panomics, panomics is actually the integration of the omics data sets together may provide a holistic view of disease pathophysiology. And also this panomics also can be used to select, of course, it will be used to select the best medication for the treatment. So again, what we learn about the panomics for the precision medicine, like the multi-omics data sets, and then we have from different kind of omics data set, and then we use the different kind of methods and then application, and then we will see the biological insight of this all of this data, and then yes, we can come up with the precision prevention and treatment for hopefully for each individual. So just a little cartoon to show why panomics is important. Just like when we learned about the single omics, it's something like, we see an elephant, but only with the we can see only a part of the elephant, not the whole system, not the whole elephant. So for example, we see the genomic data, so we can only have this part of the whole system of the individual. So also when we see we get the epigenomics data, and probably it's only this part of the elephant system. However, when we when we study all of these system, and we come up with the multi-omics, and then we integrate this data, and then we come up with the panomics, which is the integration of multi-omics. So it is the integration of all the omics data to come up with the what is called the integrated data. So and then precision medicine is so important. And I will state us out again that This is prevention and intervention. Yes, it can be tailored rather than using the same approach for everyone. As we receive our everyday treatment today, so hopefully by the precision medicine, it used to be called personalized medicine, but however, the term is moved to the precision medicine, and because it is about the precision treatment for everybody, not the personalized for everybody. However, it's a precision treatment and prevention. So because using the precision medicine approach, and then the patient will be will have a better result of the treatment after receiving the appropriate care at the appropriate time. So this also will save, hopefully, will save some fund and making healthcare more efficient and more sustainable. And then and this is also expected to have a positive effects on the patient in terms of reducing the likelihood of negative side effects because it is tailored for everybody. Some maybe only one patient may only need a very small amount of drugs compared to the other person, maybe patient A for the same disease. Patient A only needs a very small amount of drug to be responsive, and then the other person will need a higher amount of drugs in order to respond to the drug and to cure the disease. So let's see with the quantitative traceability of each person. Then when we see here genetic variants, for example, some SNP, although SNP it's not related directly to to a disease, but SNP single nucleotide polymorphism, it will also probably it will determine our predisposition of a disease. So when we talk about the QTL or quantitative trait lucky, it is a specific region on a chromosome which harbor genes that controlling the traits. And what happens when we'll see when we individual has a SNP genetic variation in a gene, so it will also affect the its transcriptome and also the epigenome and transcriptome and proteome and finally the metabolome, and this all integration of these omics data, and then we'll see the potential regulation among different omics data, which is indicated by arrow, and then this manifestation, the word cloud for the manifestation of the QTL in human disease. Like we'll see the bigger the word cloud indicates the relative percentage of the reports of QTL and related disease. So this is really a complex system. Then it cannot be when comes to a disease, it cannot be separated one another. So it has to be studied in the whole system. So so I talk about the biomarker that as everybody has because of this different genetic makeup of that we have. So a biomarker or a biological marker is a measurable and quantifiable biological parameter that serves as indicator of a specific physiological state. For example, when we have one kind of disease, and we will have one biomarker. So every disease have a different kind of biomarkers. So when we have this asthma, and it we will have the different biomarker for asthma, and then when we have the probably when we have the we are infected by SARS-CoV-2, for example, we will have different kind of biomarker too, and then based on this, the doctor will and then we'll come up with a decision on what kind of treatment we will receive later on. So and then according to what health organization, a biomarker is almost any measurements reflecting an interaction between a biological system and a potential hazard, which may be chemical or physical or biochemical. So why a biomarker is important? So it is, for example, we see the biomarker for diagnostic, for example. So it is to confirm the presence of disease and maybe subtypes or medical condition. And when we talk about the biomarkers for monitoring, and it can be used to assess the present status or the disease or medical condition, and to evaluate the response to the intervention. So we will have a very diverse of biomarkers for each individual, and also for the every disease manifestation. So that's why because of this very diverse of biomarkers, and then there is a need for panomics, which is the integration of all the data, and then it will come up with the signatures of the individuals, and then eventually it will be used for tailoring any medication of any individual for having the tailored therapeutics. So let's see, for example, the use of the panomics biomarker for reducing the mortality from ovarian cancer through an early detection. So we can go to have our biomarker assessed just not after having diagnosed with the disease, but however, we can also have it before, just like, for the prevention, for also for the early detection of the any kind of disease. As we see, usually cancer is somehow a very deadly disease with the worst prognosis, and then it has a very highest mortality rates, especially the ovarian cancer. So the the onset of the cancer usually very late. So there is a lack of specific early symptoms of the ovarian cancer, and then so the patient usually come see the doctor at the later stage. So based on this, there is an urgent need for improved biomarker for ovarian cancer, and for the use in research and also in their clinical practice. So usually, when the patient go to see the doctor, then the stage is already late, and then based on the biomarker, and when we acknowledge that we are prone to cancer, for example, because it runs in our family, so it's better for us to get ourselves to the discover our marker, for example, and then we will go through the risk assessment and also screening, and based on this, we will have the prevention. For example, if somebody is prone to ovarian cancer, maybe they will have a choice to go to the their ovarian removed, and then also maybe for somebody who is prone to the mama cancer, and then they will probably have a choice to get the mama removed. It's an option actually. It's somehow, and then patient will be suggested to have the some option for the prevention. However, it's even though we run the risk from our family, not all of us will eventually have the disease. That's why this we need the risk assessment based on our biomarker discovery, and then also our physical data. So yes, back to the ovarian cancer, and then for example, we will have the part of we will have our sample taken from many parts of our body, for example, it's from our tissue and blood, and also ascites, and probably uterine lavage, urine, and cervical smear, and then these the biopsy samples, and we will go through these omics assessments, and then we will have the multi-dimensional data, and also as I mentioned earlier, it's also the data will include the physical symptoms, clinical data, and also the environment data, and our lifestyle also. And thanks to the machine learning approach, and then this all data will be integrated, and then will be validated. So we will have the insight into the pathogenesis of the ovarian cancer, for example. So these all data will be combined, of course, using when we learn one by one the omics data, it's really impossible to come up with the conclusion. However, we will need a machine learning to, to come to the validation for making a the very right decision. So this when we have this one or more omics information, and then we'll have also the pan cancer classification, and then we will have the all our information, and then we will later on we will get a treatment. And also this, after getting a treatment, also we will have the our omics data to see whether this treatment is effective or not, and then it's this multi-omics interpretation will capture the meaningful clinical feature of every individual. So it is more or less the same. Yeah, so we're using the machine learning approach to classify the disease manifestation. So after getting treatment, so we also need the drug response profiling, and then combined with the proteomics data, because the I will propose this probably I will show here this. Okay, this when we have this disease etiology, and then we will have the determination of the cause of disease or the pathology, and then we will have our panomics data integration, and then it will be assessed. And also for the precision, for example, this is a healthy individual, and it has panomics information, and then it will be integrated in the lifestyle and environment data, and it will help this with the precision prevention. However, a sick person, for example, come, and then it will the clinician or the doctor will use the disease etiology, and then it will get the panomic information, and then it will the person will get a treatment, and then after treatment there will be a drug response examination, and it also come up with the panomics information about the drug response, and then all will be analyzed together, and then go to the precision treatment tailored for each individual. So however, there is a we will see the challenge of the panomics in the realm of the precision medicine. So hopefully, with the advent of precision medicine, there will be a prospects for the better and more targeted healthcare in the future. This seems so promising, although of course it will needs very huge amount of information for each individual, and so because of our genetic makeup, and then environmental factors will work together to bring out the disease, or also our predisposition to the disease, and then also we will see that the artificial intelligence is very helpful in the interpretation of omics data. It means that we will have to have the very high high performance computing tools for this, and then because analyzing one data is very very robust, and then it will have the very it will have the this big data of each individual will need a very high performance computing technology. So and then not to mention also the data standardization to enable the multi-modal integration, and then also the AI supported drug modeling. And we need to consider also the data privacy and regulatory aspect, and also the economic implication. And yes, all will come the policy of the data sharing, and also the integration for other omics technology. And however, this because of the promising panomics data for the precision medicine, and I think it is very inevitable to go further for this the precision medicine. I think that's all that I can share with you today, and thank you very much. And now I am giving I'm back here the time to Pak Yusuf as a moderator. Pak Yusuf, you're still mute.
Oh, okay. Sorry. Yeah. A very nice presentation, Prof. Trina. I still remember when I watch some movies, in the future, the doctor is accompanied by this machine. So whenever the patient comes, the doctor will just take a little sample, probably from the sweat, probably.
Yeah.
And eventually the machine will analyze everything using the AI, and eventually it will give some prescriptions or suggestions to the doctor which drug that's suitable for that particular person, because as you mentioned, every people is unique in terms of genes, right? So yeah, this is very interesting. And now we open the question from the floor. So now we have Pak Jacob from Fakultas Biology KSW. Yeah, please, Pak Jacob, you can ask question directly to Prof. Trina.
Thank you very much, Professor Trina. I would like to ask, in practical manner, how long to get precision medicine for the patient to be cured? Yeah, because before I have experience with my close friend who also have breast cancer. It is very difficult to be cured. Yeah, the same medicine and different result, and then she eventually cannot get to be cured and then die, something like that. Yeah. Thank you.
It is if we talk about how long actually when we come up with this precision medicine will arrive to Indonesia, and I don't know, it will take as you know, the Kementerian Kesehatan has now initiated what is it called, BSG. It will collect all information about the clinical data of the patient, and also the genome information of all patients. But it just recently been launched. But maybe outside of Indonesia, it has already been launched. Because the advance of technology. For example, in the Western countries and the European countries, they have very advanced technology, and also they have a support system that will speed up the precision medicine things. But I cannot say anything about, when the precision medicine we can have in Indonesia. Yes, in Indonesia. However, the Kementerian Kesehatan, the Ministry of Health, has already initiated that, in the hope that otherwise, if we hasn't initiated that, we will not come to the, we will not catch up this this technology. So, we hope. We don't know what we do hope that it will come maybe in the last 10 years or sooner. I hope so. So I cannot answer your question precisely. I'm sorry, Pak Jacob.
Yeah, thank you, Bu.
Yeah, my pleasure. You're still mute, Pak Yusuf?
Oh, yeah.
Sorry. Yeah, sorry. Yeah, now we have the second question from Ishfaq Khan. Are you here? Can you open mic to ask question directly to Prof. Trina? Okay, so I will ask. I will read the question. So he aware that there will be a large number of measurements and the limited number of test subjects. Unique problems arise in omics studies involving statistics and bias.
Yeah.
How to deal with that?
Okay, so obviously it will not be, this panomic strategy for precision medicine cannot be handled only by molecular biologists. However, it will also with the help of a statistician and machine learning technologies and all this, not only of the software, also the hardware, also this, and also the policy altogether. It's not a standalone system. It is an integration system, so it will cover all many expertise in this panomic strategy. So, as I mentioned earlier, it is very complex things, but if we don't start, so we will always left behind. So
Okay. Yeah.
I think that's it.
Yeah. Thank you. Yeah, I think the last question will be from me, I guess.
Yeah.
Yeah. About this, you said that if we don't start now, we will always left behind. And as we know, to implement this into the clinical applications is still a huge challenge. But do you have any example that we already started the collaborations with a hospital? Let's say that we study from a certain biomarker in the gene, and then we studied in the proteomics side and correlated with the phenotype. So in conclusion, we could say that, oh, so if the person have this biomarker, it could lead to some disease that is not happening now, or as a early diagnostics. So have you started this type of collaborations with the collaborating hospital?
Yeah. Let me show this this information about the yes, actually Kementerian Kesehatan has launched the BGSI initiative national, the first initiative national for the detection of the potential disease in the future. So you can see here the information that this is a collaborative agreement between several hospitals and then several university and the this biomedical and genome science initiative from the Health Ministry of the Republic of Indonesia. It will serve as a collection of the information that can be assessed by all clinicians and also scientists, and it will be a portal, or the first portal for the information of the genomic of Indonesian all genomic sequence of an Indonesian individual, and the, together with the clinical data. So yes, we have already had this, and it is based in the Ekman building, Cipto Mangunkusumo Hospital. So yes, we have already.
Okay, thank you. Yeah, so this is interesting because this is also one of the goals of MABI, yeah. And now, alhamdulillah, we already have the BGSI. And Prof. Trina, since you are here in Bandung, if you have time, we'd like to invite you actually to our Congress, and also we can having further discussions about this. And later the committee will tell more in detail about the Congress this afternoon. So please feel welcome if you have time. We would like to invite you, and also we would like to invite all the MABI members to the Congress. Okay, we are running out of time, and also there is no other question from the floor. So we would like to appreciate for the talk from Prof. Trina. Give big applause to her. Thank you so much. And I think this presentation also is related with the next speaker, Prof. Bimo. Okay, Prof. Bimo, I thought, oh, okay, so you are already here. So welcome, Prof. Bimo. So I would like to read a short resume of Prof. Bimo. So Bimo Aryo Tejo, PhD. So he graduated from the University Putra Malaysia in biotechnology. And currently he is an associate professor in Department of Chemistry, the University Putra Malaysia. And he has 58 papers published until now, and one of the recent one is on the rational design and characterizations of short antifreeze peptide derived from nolium-parent antifreeze protein. And yeah, I remember that his expertise is one of the pioneer in structural bioinformatics, I guess. Yeah. And without further delay, I would like to invite Prof. Bimo to deliver your talk about the therapeutics peptide, I guess. Yeah, in discovery. So please, the floor is yours.
Alright, okay, thank you Pak Yusuf. Suara saya jelas ya?
Ya, clear.
Slide saya kelihatan ya?
Ya. Oke, perfect.
Right. Okay. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh, and good morning. So today I'll be presenting one of the works that I've been doing for the past 15 years. Yeah, it's about rational design of therapeutic peptides. And as Pak Yusuf mentioned just now, currently I'm working at the Department of Chemistry, University Putra, Malaysia. So in my work, I combine experimental work. I did a lot of synthesis in the lab, and in designing the molecule, I use computer. So I use computer, and then I synthesize the molecule in the lab, and then I test the molecule using various biophysical techniques, for example, NMR. And so actually it covers almost everything. Yeah. So if people ask me, what is my research interest? Is it computational biology? I'm a bit confused to answer, because to me as a scientist, we are okay to have a wide research interest, especially if we are working on a multidisciplinary research. So why peptide is interesting to me, yeah, at least to me, yeah, because when we look at this data from last year, this is the drugs approved by the FDA last year, classified on the basis of their chemical structures. You can see that the number of peptide drug approved by FDA is more than natural product. I'm not saying that you have to stop doing natural product. I mean, because I know that there are still a lot of potential in natural product research, but as you can see here, that peptide is getting more and more popular. We don't combine it with protein enzyme yet, but as you can see, the number of peptide is more than the number of natural product approved by FDA last year. Yeah. If you talk about peptide 30 years ago, people will laugh at you. Nobody believe you if you talk about peptide 30 years ago, because peptide is not stable. Yeah, when you make a peptide as drug, you take a peptide orally, it will be destroyed in your stomach, yeah, because peptide is not stable. And then I'll show you some challenges in designing a peptide for drug. But now we know a lot of ways to stabilize the peptide, and it's not an issue at all. So that's why the number of peptides approved by FDA is getting more and more every year. And if you look at the the future of therapeutic peptides, 2 years ago, the global peptide drug market, it was more than $36 billion, and it is predicted to increase in 2030 to be estimated $66 billion. And currently there are more than 200 US FDA approved peptide and protein on the market, and 500 therapeutic peptides are in preclinical development. So actually the future is very bright. Yeah. The future is very bright. So this is one of the reasons why I'm interested in peptide research. Basically, it's not about money, even though I show you a stack of money here, actually it's not about money. But one thing that makes me interested in peptide is how easy to synthesize a peptide. Because peptide is amino acid, it's a polymer of amino acid. So we have 20 amino acid. Natural amino acid, and we can combine them in many ways, and by using this technique introduced by Bruce Merrifield, we can synthesize a peptide in an easy way. We call this technique as solid phase synthesis. Previously, the peptide has to be synthesized by solution phase. So you put all amino acids in a solution. You let the reaction happen, and then at the end you have to purify the peptide, which is very difficult. Yeah, but Bruce Merrifield, in 1970, he came with the idea of doing peptide synthesis using solid phase synthesis, which simplify the synthesis. It cuts the number of purification that we need to do, so that even non-chemists like my student, he's not chemist. Yeah. He's not chemist. He can do the synthesis. He can get the peptide within 2 weeks. This is my student. Now he's a doctor. Yeah, he got his PhD already. Dr. Izzuddin. Now he's working in one of the biggest university in Malaysia. So he synthesized the peptide within 1 or 2 days using this peptide synthesizer machine. And then it is very easy. Even if you are not chemist, you can do the synthesis. You just put all the amino acid based on the sequence that you want in the machine, and you leave it for 1 or 2 days. You can go for vacation basically 1 or 2 days. Hopefully, the machine does not leak. Yeah. So after 1 or 2 days, you come back, you harvest the peptide, and you purify using HPLC, and you get the peptide within 2 weeks with high purity. Yeah. So synthesizing a peptide is easy. Yeah. So it's good to those who hate organic chemistry. They don't like doing complicated synthesis. So peptide synthesis is one way to go. And another good thing about peptide is even as short as 3 amino acids, it can have activity, biological activity. Yeah. This is one example how a peptide, a short peptide, as you can see here, the length of the peptide is 3 amino acids. We call this peptide is RGD, yeah, arginine glycine aspartic acid, discovered by Bisbacher and Roslati in 1984. Now people use this peptide as anti-blood clot, yeah, as a drug. Yeah. As you can see, the structure is very small, only 3 amino acids. Only 3 amino acids, yet it has a biological activity. Or you can synthesize a long peptide. For example, this 36 amino acid peptide. It's very easy to synthesize. Yeah, it's a linear peptide. All the peptide they use is natural peptide, and this is drug, fusion. And the cost for treatment per year is $25,000 US. So peptide is very versatile. You can synthesize a short peptide. You can synthesize a long peptide. Whatever it is, your imagination actually is unlimited. Yeah. Of course, it comes with some challenges. Yeah. The first challenge is how we choose the sequence. As I told you, you can synthesize as short as 3 amino acids, as long as 40, 50 amino acids. But the problem is how to choose the sequence. And also peptide stability. As I mentioned to you earlier, that 30 years ago, when you talk about peptide drug, people will laugh at you. They don't believe you because peptide has an inherent problem with stability. And also how to characterize the biological activity, because peptide is not an enzyme. We cannot simply do enzymatic assay to get the some information about how this peptide binds, where and how. Yeah. So this is the inherent issue with peptide. Peptide can be easily degraded, especially when you take it orally. Yeah, when you take it orally. So it will be degraded by protease or peptidases. Yeah, it breaks into amino acid. And even when you try to inject it, oh, I don't want to give the peptide drug orally. I just inject. Inside our blood, it composed of 44% red blood cell, 1% white blood cell, 55% plasma, and more than 120 different proteins, including peptidases. So you inject peptide in the blood, it will be degraded quickly. So that's the issue with peptide. So this is the list of enzymes that can degrade peptide. Yeah. So we have endopeptidases, we have exopeptidases. So yeah, it's not easy. Working with peptide is not easy, but we find a way to stabilize the peptide. Yeah. One of the ways is by using liposome. Yeah. For example, polyethylene glycol. Yeah. So we put the peptide inside the liposome, so the peptide is protected against degradation. And this is the same technology used by COVID vaccine. For example, Pfizer and Moderna, they use the same technique because mRNA is very unstable, very unstable. So they use PEG to protect the particle. Or any other polymer nanoparticle that you can use, for example, polylactic acid, polylactic glycolic acid, polysalic acid. So there are plenty of choices that we can use to stabilize the peptide. Or we can stabilize the peptide chemically by changing the structure. For example, by doing methylation, we replace with D amino acid, but D amino acid is expensive. Yeah, 99% amino acid are L amino acid. We change with D, so the peptide becomes extremely stable. But remember, D amino acid is expensive. Or we can change the peptide bond into amide bond. So, so it will become more stable, and then we can make it as cyclic as well. This is what I did in my research. I cyclized the peptide, and the peptide is more stable against degradation. Yeah. All right, so let's go to what I have done for the past 15 years. So this is the first work of what I have done with peptide. So I synthesize and I design and synthesize ICAM1 derived peptide. And this is the reason why we work on this peptide, because of this T cell activation and immunological synapse. So this is the how T cell is activated. And in most cases, we need our T cell to become activated, yeah, especially when you're infected by virus, when you are infected by bacteria, when you take vaccine, you want your T cell to be activated. Yeah, you don't want your T cell does nothing. Yeah, it will be detrimental if your T cell is not activated and your T cell does nothing if you are infected by virus. Yeah. But sometimes we need to reduce the activity of T cell. Yeah, when we deal with autoimmune disease, because in autoimmune disease, our immune system attack our own tissue. So in that case, we want to reduce the activity of T cell, yeah, so that we want to stop the T cell activation. Yeah. So in this case, we designed a peptide that we hope can block the interaction between ICAM1 and LFA1. Yeah. So ICAM1 and LFA1 is called as the signal 2, yeah, that happens after the MHC peptide MHC complex interacts with T cell receptor. That's signal 1. So signal 2 happens after signal 1. Yeah. So we try to stop that. Sorry. So this is the peptide that we try to design. So we did docking. We see how the interaction between LFA1 and ICAM1. Yeah. So ICAM1 comes from the antigen presenting cell, and LFA1 comes from T cell. Yeah. So this is how we design the peptide. We did docking to see how ICAM1 interacts with LFA1, to see which peptide actually interacts both from LFA1 and from ICAM1. And these are the peptides that we design. Yeah. CIBR, CIBL, CLBL. Yeah. So the peptide that we design and we synthesize comes from this area. Yeah, we call this peptide as IBR peptide. Yeah, so this is the sequence, PRGG, SVLT, TGC. And we put penicillamine at the N-terminus because we want to cyclize this peptide. Yeah, so we synthesize the peptide, we cyclize the peptide to increase the stability of the peptide. So this is how the peptide looks like. So we solve the structure of the peptide using NMR. So it adopts beta-turn structure, exactly like what we saw in the crystal structure. In the crystal structure, the peptide has beta-turn structure. And after we synthesize the peptide, it still maintains the beta-turn structure at PRGG sequence. So we are happy with this. And then we want to know how this peptide interacts with the LFA1. Yeah, so we express LFA1. Yeah, and we incubate with our peptide. Yeah, and it turns out that our peptide actually can bind to LFA1 at the site that we never predicted before. It's quite interesting. We published a paper a long time ago in 2007. So it turns out that it binds to this area. Yeah, to this area, yeah, at the bottom of the LFA1. So we try to shorten the peptide, yeah, because what we need from this peptide is only PRGG sequence. So we thought, well, what happened if we shorten the peptide? Yeah, instead of using PRGG, SVLT, TGC, we shorten it into 7 amino acid, yeah, CPRGG, SVC, to see whether this peptide works as the parent peptide. Yeah. We published on the paper a long time ago in 2008. And this is the structure that we solve using NMR. And it shows us that it has similar structure as the parent peptide. Yeah. And then this is docking. We want to know how this peptide interacts with the LFA1 and it binds to, we call it as L site at the bottom of the LFA1. And it hinders the movement of alpha helix 7 of LFA1. We did some cell culture work as well. We published in long time ago in 2009. And it shows that the peptide enters the cell through energy-dependent mechanism, not through passive diffusion. As you can see here, that when we put the cell at 4 degrees Celsius, nothing happened. means the peptide cannot enter the cell, but when we increase the temperature to 37 degrees Celsius, you can see that the peptide enters the cell. So it's energy-dependent mechanism through LFA1 receptor. All right, so that's my earlier work about peptide. And then this is what I started in 2010. Yeah, in 2010. I worked on a different peptide. And this peptide is designed to interact with another enzyme called PAD4. And this is the disease that I was dealing with, is rheumatoid arthritis. So rheumatoid arthritis is autoimmune disease with unknown etiology. It attacks people in any range of age. I had a student, still young, 20 years old, but she got rheumatoid arthritis. She did her PhD with me on this topic, on the topic related to her disease. So amazing. So she fight real hard to finish her PhD, working on disease that happened to her. And she got the PhD after 5 years. So still young, but she got rheumatoid arthritis. So it attacks people in any range of age. So current treatment, they are mainly focused on pain relief. Yeah. So no drug that can address the root cause of rheumatoid arthritis. So we need a new drug. So this is the mechanism. So basically the mechanism is because of this citrullination process. Yeah? So we have an enzyme in our body called PAD. This is the enzyme. And the job of this enzyme is to convert arginine into citrulline. When you look at carefully, it has positive charge here in arginine and it has 0 charge here in citrulline. So this process is a normal process. Ya, biasa di dalam badan kita ya. It's a normal process. And this process happen as part of the immune mechanism. So if you're infected by virus or bacteria, this process will happen. Yeah? But the issue is if PAD is overactive, yeah, it's overexpressed, overactive, so we have too many citrullinated protein. Ya, itulah masalahnya. Ya, kalau kita ada protein tercitrulinase yang terlalu banyak, ya, then our body will see this protein as an enemy, musuh. Yeah. So that's why our own immune system attacks our own protein. Then this is how rheumatoid arthritis happen, because of our immune system attacks our own protein, which is citrullinated protein. So we design a peptide, yeah. So we design a peptide using a substrate of PAD4, nucleophosphate. So there are 8 peptides that we synthesize based on the sequence of nucleophosphate. We choose the sequence with arginine in the middle of the sequence. Ya, jadi ada 8 peptide yang kita buat. So, and then we test how this peptide interacts with PAD4 experimentally, yeah, not by doing docking or whatever. And of course, we have to express the protein. So a lot of work here, expressing the protein, synthesizing a peptide, testing the peptide using enzymatic assay, solving the structure of peptide using NMR, testing how the peptide interacts to protein using biophysical techniques. So a lot of work here. So from here, we can see that peptide 5 actually is the best peptide among these 8 peptides. So we choose peptide 5, and then we try to shorten the peptide 5. Yeah. We try to shorten the peptide 5. So we can see that if we shorten the sequence into 5 amino acid, yeah, 5 amino acid, yeah. So it turns out that the activity is better than the parent protein. So we choose this modified peptide and we modify the sequence. So a lot of synthesis work here. So this is the peptide that we synthesize. And the arginine of the peptide, we change it into this functional group so that it can attack the active site of PAD4. Yeah. Why we put that group, that functional group, is a long story. Yeah, it's a long story. But to make a long story short, we choose this group because from our previous work, we saw that this group can attack cysteine 645. Cysteine 645 located at the active site of PAD4 enzyme. So this is the IC50. It's not very good, yeah, but this is peptide. Yeah? This is peptide, and normally when we work with peptide, we cannot expect to have nanomolar IC50 like we have when we work with small molecule. Yeah? If we work with small molecule, we can get IC50 around nanomolar scale, but this is peptide. Normally the IC50 is not very great, but that's fine. And then how it binds with PAD4, we did a kinetic study, and we can see that it's a reversible and competitive inhibitor. We also solved the structure of this peptide using NMR. So we did 1D and 2D NMR spectroscopy, and we can see that it has a random coil structure. And we use this structure to do molecular docking. And this is how the peptide docks to PAD4. And exactly like we predicted, the furan ring of the peptide enters the active site and interacts with cysteine 645. So that's why we are quite happy with this result. Exactly like what predicted. And then this is the last, yeah. We did this work in 2015, 2014, yeah. So right, so this peptide is actually, we designed the peptide as an inhibitor, yeah, as an inhibitor of hepatitis B virus. And the reason why hepatitis B virus is still a major issue now is because of expensive antiviral drug. Only 20 or 30% of this drug shows sustained virological response after withdrawal of the substance. This is the same issue with Paxlovid, yeah, COVID drug. When you stop Paxlovid, the virus rebounds. Jadi masalahnya sama, ya. This is what happened with Paxlovid. So you stop Paxlovid, the virus rebound. So and then the same thing happened with antiviral for HPV. When you stop the drug, on only 20 or 30% show sustained virological response. And prolonged therapies with nucleotide analogs result in drug-resistant mutants. And this is how the virus looks like. So we designed a peptide that can inhibit the life cycle of this virus. And first, we need to know the anatomy of HBV. Yeah. So this is the anatomy of HBV. Yeah, it has a spike. Yeah, a lot of spike. Yeah. If you see spike, you automatically remember about SARS-CoV-2, yeah, virus. So you can see spike. Yeah? We call this as LSBSAG. And then merah M SBSAGs as well. And then S SBSAG. And then if we look into it, yeah, more detail on the structure, this is a preformed nucleocapsids that will interact with virus or waste protein, and this interaction is very critical for the HPV life cycle. So we want to stop that interaction from happening. So we want to design a peptide that will stop the interaction between nucleocapsid, yang kuning ini, with the surface protein of the HBV, hepatitis B virus. So the peptide that we design is taken from pre-S1. Yeah, this is the peptide that we designed. Sorry. Yeah, so this is the, sorry, it's too fast. These are the two peptides that we designed. Yeah. First peptide is taken from pre-S region of LSBSAG. The second peptide is taken from the S region of LBSAG. And then we did NMR spectroscopy to see the structure of the peptide, as well as to study the how the peptide interacts with the nucleocapsid and also with the surface antigen of the peptide or of the virus. And then we did molecular dynamics, okay, and then molecular docking as well to see how this peptide interacts with the target. So this is the interaction study. We did STD NMR, saturation transfer reverse NMR, to see how the peptide interacts with the target and combine with the molecular docking. This is a peptide. This is a one-dimensional NMR spectrum. Yeah, and the assigned peak are the peak from amide, NH, because when we do NMR, especially for peptide, the first peak that we need to find is the amide. Yeah, we need to make sure that all the amide peaks are there before we solve the structure of the peptide. So this is the first peptide. This is the second peptide. So we are able to locate all the amide peaks on the NMR spectra. And then we did 2D toxi, total correlation spectroscopy of peptide 3S. And then this is peptide S. And this is the structure. Yeah. When you do NMR, you will get an ensemble of structure. You don't get single structure. You get ensemble. Why? Because when you do NMR, your protein or peptide is in solution. So your peptide or protein is dancing, it's moving. Yeah. You cannot see this in X-ray. In X-ray, you get only single structure. So this is the beauty of NMR. You get an ensemble of, of structure. So you can see how the peptide moves. Yeah. And then we did docking. Once we get the structure from NMR, we use that structure to see how this peptide interact with HBCAG. And the way it interacts is amazing. It docks into the pocket of the HBCAG. And then this is STD-NMR. STD-NMR is one technique in NMR to see which amino acid, which residue in the peptide that interacts with the protein. So we can actually find a consensus between molecular docking and STD-NMR. Yeah, so that's what I have done. And of course, for future work, yeah, we can use the similar technique to design other things. Yeah, and one of the main concerns now is a drug and vaccine for COVID. This is something that I'm not doing it now. Yeah, I'm not doing it at the moment. Yeah, but this is maybe among the audience. This could be your main concern. Yeah, because until now we have only one effective drug for COVID, yeah, which is Paxlovid. Molnupiravir, forget it. Molnupiravir is not effective. Remdesivir, the issue with remdesivir is it has to be given in a hospital setting. So the drug that can be taken orally at home is only Paxlovid, and Paxlovid has an issue with viral, the virus that rebounds. So this is still an existing challenge for us to find new drugs for COVID and vaccine. Of course, we are dealing with new variants now. We come up with updated vaccine, but still, the updated vaccine, because of the issue with immune imprinting, yeah, the new vaccine is not as effective as we expect. Yeah. But this is an indication that there are a lot of things that we have to do. Yeah. And one of the things that you can do, yeah, especially young scientists among the audience, yeah, you can design a multi-epitope peptide-based vaccine against SARS-CoV-2. This is not a new idea, yeah, but this is something that you can do, yeah, for the future to expect to deal with the appearance of a new variant and subvariant. Okay, so these are the people who are involved in this project. Yeah, Professor Sian from US, and then from UPM, Dr. Teo is my student. Dr. Zalika and Dr. Izuna also my student. Yeah. Now they become my colleague now. Dr. Azira is my student and also my wife. Yeah. So both of us are scientists, yeah, and we publish paper together anyway on hepatitis B virus. Now she's working on leptospirosis vaccine. And Malcolm and Stephen and Busan are from Edinburgh, and Pedro is from Imperial College. So that's all from me. Over to you, Pak Yusuf. Thank you. Assalamualaikum.
Waalaikumsalam warahmatullah. Thank you, Prof. Bimo. It's very interesting talk. Yeah, and also it's always been delightful to see eventually the molecule is really working in the laboratory, and your slides really inspiring us that the idea of molecular modeling is also to help the experiment to have a better result. And yeah, congratulations for that. So we already have a few questions from the floor. The first one is from Umi Baroro. Do you want to deliver the question by yourself? Okay, she's here actually in the room. Okay, so yeah, it's very nice presentation. So there are 20 amino acids as we know, and she asked how to design a peptide that can inhibit the disease, maybe specific to some disease. And for docking study that you did, which software that you usually use? Because probably because the peptide docking is quite tricky, because there are a lot of degree of freedom, flexibility, and things. So yeah.
Yeah, all right. Very interesting question. Yeah. First, yes, we have 20 amino acids, and the challenge is to get the length of peptide that you want, that can inhibit the disease. Of course, you have to know the target. The target. Yeah. For example, when you design a peptide for vaccine, it has to interact with major histocompatibility complex molecule, right? Either MHC1 or MHC2. When you look at the structure of MHC1 and MHC2, they have a very small groove. Ya, kayak ada lembah itu tapi agak kecil. So the peptide that can enter that is only from 9 to 15 amino acid long. Yeah, only 9 to 15 amino acid long. Yeah. If you design a peptide for anti-blood clot, like I show you in the beginning of my presentation, you can design a peptide as short as 3 amino acid because the receptor is very small. Yeah. If you design a peptide for example, for HIV, yeah. When you look at the structure of HIV protease, actually it's a dimer, and in the middle there is a tunnel. So you can design a peptide with, longer sequence. So you have to know the receptor, the structure of the receptor, the three-dimensional structure of your receptor, to know the length of the peptide that you need to synthesize. Yeah, that's one. Second, for docking, we cannot use autodock-Vina. If you're dealing with peptide, normally we use HADDOCK. Yeah, HADDOCK, HADDOCK. Yeah, because that's the peptide so far. That's the software that so far, as far as we know, is the most suitable for peptide docking. Okay, so we cannot use autodock-Vina. We use a HADDOCK in our group.
Okay. Yeah. Thank you. So the second question is from Irfan Nurullah. So he wants to know about the stability of the peptide and how to determine it, and then maybe also the experimental things probably to check the stability of the peptide.
Okay, so most peptides are not stable. Yeah, most peptides are not stable, but we can try to increase the stability of peptide by doing, as I show in my presentation, by doing some chemical modification. Yeah. But my concern actually is not only about the stability. The first that we need to be concerned when we design a peptide is solubility, kelarutan. Yeah. So this is the first thing that we need to keep in mind when we design a peptide. Stability, it comes later. Yeah, we don't have to worry at the beginning of our peptide design because we know most peptides are not stable. We know that. Okay, but the first thing we need to keep in mind is whether it's soluble or not. We can predict. Yeah, we can predict the solubility of the peptide. There are plenty of software that we can use to predict the stability, because when we synthesize the peptide and we use the peptide in our project, in our research, the peptide has to be soluble in water, right? If the peptide is not soluble, there is no use of using that peptide. So first is solubility. Don't worry about stability. Most peptides are not stable anyway. Yeah. And after you synthesize the peptide and it's soluble, and then you use the peptide for your experiment, either in vitro or in vivo, then we know the stability. Yeah, by doing PKPD, yeah, pharmacokinetics, pharmacodynamics, yeah, we know how stable the peptide is. If we know that our peptide is not very stable, then we try to increase the stability. If modifying chemically is not possible, you can put the peptide in a vehicle like liposome, you know. So that's how we deal with this stability issue. But don't worry, in the beginning of your project, don't worry too much about the stability of the peptide.
Okay, thank you. So while we're waiting for the next question, maybe I will like to chip in into that one. So yeah, about the stability, because when we read from some literatures, usually people, when they develop the peptide for drug or for maybe vaccines or things, they usually use some what do you call it, the carrier, the delivery agent, yeah. So, and in your presentation, I think there were some studies on the internalizations of this peptide into the cell, if I'm mistaken. So, what is the future plan for that? So, you will use the bare peptide, or you plan to use some carrier in the future?
Okay. So we did that work to see whether the peptide enters the cell by passive diffusion or by energy dependent. Yeah, because if it enters the cell by passive diffusion, it means the peptide does not interact specifically with the receptor that we want. Yeah. But our result shows that it interacts with the receptor that we want. It enters the cell by energy dependent mechanism. Yeah. So in the future, so we still use the peptide without any carrier anyway.
Okay.
Yeah, without any carrier, because for that peptide at the moment we just did it for in vitro and in vivo assay. Ya, kita belum lakukan human studies. Ya. When we go into human studies, then we might have to protect the peptide with some liposome.
Okay.
We might have to, but at the moment we can see the peptide is pretty stable in vitro and in vivo.
Yeah, it's very interesting indeed. And I still remember also that you mentioned there is a very short peptide, the RGD.
Yes.
Yes. So the RGD also have some signaling properties. I mean to give a signaling in the growth of the cell. And so I read some studies also. This RGD can be used to treat the, you know, the gagal ginjal.
The kidney injury.
Yeah, the kidney acute kidney. Yeah, because somehow it can be used to repair the kidney cells. And
yeah, I don't know about that. It's a new information for me.
yeah. So I was thinking that if the synthesis of this peptide is easy, then probably we could think very fast how to deliver this peptide to finally reach that cell in terms of pharmacokinetics delivery. You know, so currently in the market, when they use the peptide product, so they always use this liposome, or can
Yes,
also deliver by
yes. The peptide is protected, yeah, because the peptide is inherently not stable in plasma, so they always protect using a carrier.
Okay. So yeah. Any questions from the audience? Please raise your hand. Otherwise, there will be our chit chat with Prof. Bimo between me and him.
Yeah, again, don't worry with the stability, because you remember that the mRNA that we use for vaccine is not stable. mRNA is very fragile.
Okay.
Yeah. But we somehow still use it for vaccine because there is a way to improve the stability, by putting inside the PEG nanoparticle and also by keeping the peptide in low temperature. So don't worry too much about stability. You can worry about that later on. But first, just make sure that the peptide works in vitro and in vivo.
Okay.
And also, if in Indonesia, I haven't seen so many scientists are moving into peptide research. Yeah. But this
Oh,
is something that
me.
I think this is something that very promising, yeah.
Yeah.
Because synthesizing a peptide, because I always believe that as scientists, once we design something on computer, we still have to make it, right? We still have
Okay.
to make it and then test it.
Yeah.
So
Correct.
peptide is something quite easy to synthesize. Yeah.
Okay.
Even if you cannot synthesize, you can buy. Nowadays, I just buy, nowadays,
Okay.
because buying the peptide is even cheaper than synthesizing it.
Yeah.
It's cheaper.
Indeed. Okay.
Yeah.
Indeed. Okay, we have another question from the floor, from Risa Alivianti. So what is the best formulation to optimize the use of the peptide? Probably we can use nanotechnology. Yeah, it's nanotechnology applications on the formulations of the peptide. Do you have any? Yeah.
It's very difficult question. Of course, it's case by case basis. Yeah? So, so it depends on the purpose of the peptide. How are you going to put the peptide in our body, either orally or by IV, intravenous. It depends. Yeah, there is no single answer for that. But yeah, we still need to protect the peptide from degradation by using nanoparticle. But what type of nanoparticle we use, it depends on how we deliver the drug to our body, because oral delivery and IV injection delivery, it requires different type of a nanoparticle.
Okay, thank you. Okay, please, do you have another question for Pak Bimo? While waiting, because we still have time actually, and this is very interesting topic. Yeah, when you give a talk about that some specific fragment can lead into the autoimmune disease because somehow it could come from our host genome. Yeah, I mean host peptide or self epitope self peptide. So what is your opinion of developing the peptide-based vaccine? Because one of the concern that if we use the whole antigen and this pathogen can have similarity to the human proteome, yeah, and I think this peptide-based could be useful to select those specific to the antigen, but not to the human proteome. Do you have any experience on this? And then maybe if you want to add your comment on that.
Yeah, this is very important. Yeah, when we design a peptide, we have to make sure that the peptide belongs to the pathogen. Yeah? It doesn't belong to us. Yeah? If the peptide belongs to us, then it will cause the autoimmune reaction. Yeah? So that's why bioinformatics is very important here before we synthesize the peptide vaccine. Yeah? And yeah, to make sure that the peptide we design doesn't have a peptide from human. Yeah. So we did once long time ago. I forgot that. We did once long time ago. So we tried to design a vaccine using this reverse vaccinology technique. Yeah, and we check where the sequence of the peptide, where the human has the same sequence with the peptide. Yeah. So it's very important, yeah, to check before you design a vaccine. And but in most cases, the sequence of the epitope that belongs to virus or bacteria, in most cases, it's different sequence than what we have in human. In most cases,
Okay.
to be sure, please check before you design a vaccine, because you don't want to design a vaccine and it cause autoimmune disease, right? So please check before you synthesize a peptide-based vaccine.
Okay, thank you. So it is very important. Yeah, I did my best to. Okay, this is the last question then, because maybe most people here are familiar with the therapeutic peptide. Maybe one of them is insulin, for example. Yes, because insulin is a very small protein, And somehow now there are some optimized insulin that could be shorter than the real one. So what's your opinion that if this insulin can be synthesized, so probably it will be easier to produce and could reduce the cost of therapeutics protein that people need. Yeah. So do you have any opinion about this synthetic peptide that's already in market for the use of therapeutics?
Well, it depends on the length of the peptide. Sometimes synthesis is more expensive than expressing the peptide.
Oh, okay.
Sometimes it's more expensive, yeah. Depending on what technique you use. If you use solid phase synthesis, of course, it's more expensive. So that's why people are moving into genetic engineering.
Okay.
Yeah. But if the peptide is too small, for example, 10 amino acid, you cannot express it on cell because it's not stable. Yeah. Because if the peptide is too short, the peptide is prone to degradation. We did it before. We tried to express a peptide 25 amino acid long, 25, using E. coli. We always get fragments. The peptide always degraded. We spent 2 years because we thought expressing the peptide in E. coli is cheaper than synthesizing the peptide, right? Because we do the peptide in large quantity. But we never get the peptide. What we get is only fragments. Yeah. We spent 2 years for that. And then we changed. Okay, we fed up already. We cannot keep doing things without getting the result. And then we changed to synthesis. It's more expensive, but we get the peptide.
Okay.
So the choice of whether we do the synthesis, chemical synthesis, or we do expression using cell really depends on the size of the peptide.
Okay.
If the peptide is too short, the peptide will be degraded when you try to express it in cells.
Okay. Okay. So thank you. So based on experience, even for 25 amino acid, it's better to use the synthesis, yeah,
Yes.
instead of
Yes.
protein expression. Okay. I think it's very good discussion today, and also very nice presentation from you. So let us give big applause to presentation of Prof. Bimo.
Thank you.
Thank you so much for your time and for your knowledge here. So yeah, I think that is for my session. I would like to return to our MC. Yeah, please, Ibu Risa. Thank you.
Okay, thank you very much, Professor Dr. Trina Ekawati Talei, Associate Professor Dr. Bimo Atejo, and Dr. Muhammad Yusuf. Okay, ladies and gentlemen, don't forget to all participants to fill the attendance list on the link available in the chat box. Please make sure that you have registered for the workshop, which will be held on November 10, 12, and 13, 2022 as part of the conference of the programs. Don't miss out on this workshop because it's still very rarely available as elsewhere. The topics that are available: multiomics data integration and visualization delivered with Dr. Gitanjali Yadav from National Institute of Plant Genome Research and IPGR, New Delhi, India, and University of Cambridge. On Tuesday, November 10, 2022 at 10 a.m. Jakarta time. Immunoinformatics screening for peptide vaccine discovery delivered with Biomods and GenScripts on Saturday, November 12, 2022 at 10 a.m. Jakarta time. Single-cell RNA sequencing delivered with Associate Professor Debarka Sengupta, IIIT, Delhi on Sunday, November 13, 2022 at 4 p.m. Jakarta time. And the last one is machine learning for bioinformatics using Google Colab delivered by Dr. Sonika Tiagi from Monash University, Australia on Sunday, November 13, 2022 at 10 a.m. Jakarta time. Feel free to join the National Congress of MABI, both virtual or on-site, to Gedung Pasca Sarjana, Universitas Padjadjaran, Bandung. Please follow the link on the chat box. Distinguished ladies and gentlemen, moving on, I now present Dr. Husna Nugraha Praja, SSI MSI, to lead the second keynote talk session. Dr. Husna Nugraha Praja, SSI MSI, completed his doctoral program with a thesis entitled Genetic and Molecular Analysis of the Cross-Incompatibility Phenomenon in Maize from Scuola Superior Saint Anna Pisa, Italy in 2015. Currently, his research focus on data analysis, molecular data derived from next-generation sequencing (NGS), genomics and transcriptomics, and identification and characterization of long non-coding RNA molecules. Ladies and gentlemen, please welcome Dr. Husna Nugraha Praja, SSI MSI.
Okay, good morning, good afternoon to all the attendants that probably in another time. So we will continue our conference to the next keynote talk. Here we will have Bapak Tias Hikmawan, PhD, from the he is head of Tribe Biotechnology Digital Transformations Office from the Ministry of Health Indonesia. So Pak Tias, or probably I can call it Mas Tias. Hello, Mas Tias.
Thank you.
Assalamualaikum.
Okay.
So before we start for the lectures, I would like to read some information about Mas Tias Ihsan Hikmawan, PhD. So I would like to say that Mas Tias is probably his specialty, especially in the environmental, because he is also the auditor of ISO 4001 for environmental management system, and also he has a lot of experiences in marine science, environmental technology evaluations, molecular biology, and bioinformatics. And now I guess Mas Tias is the commander of head of the first biobank, also the biorepository for the Ministry of Health in Indonesia. So nowadays, since January 2022, Mas Tias ini established bioinformatics system for the first Indonesian Human Genome Project, the 10K project, as part of MOH initiative to support precision medicine, and also provide consultation to 7 national hospitals to develop their capacity for analyzing genomics data. And the third is the develop biobank and bioregistry information system for biomedical genomic science initiative, or BGSI, from the Ministry of Health. So I guess we can't have a lot of information, especially because this is new, and the kickoff, the launchings already, I guess, in the last August. So for Mas Tias, you will have around 40 minutes to present the informations, and we will have 10 minutes for QA sessions. Okay, please, the screen is yours, Mas Tias.
Okay, thank you so much, Pak Husna. I hope that my voice is clear.
Yes.
Is it clear?
Yes.
Okay. So let me share my screen first. So I would like to share the screen. I do hope that now you can see in the full screen of a slide. Is it showing? Is it showing, Pak Husna?
Yes, yes.
Okay.
But not
Thank you so much.
Not
Yes,
in
hello?
the preview, not in the slideshow mode.
Okay.
You should duplicate, I guess.
What about now?
Yes.
Okay, now it's showing, yeah?
Yes, sir.
Yes, perfect.
Yes. Thank you so much. Good morning, everyone. I hope that you are doing well. And thank you for having me. It is my pleasure to join in this conference. try to discuss what is the future of biomedical and genomic technology. So I would like to share a glimpse of the initiative by Ministry of Health. And I would like to start with the ministry focus to transform the health information system. And it is based on the six pillars of the transformation. The first is the primary services, as we know, like Puskesmas and Posyandu. Secondary services like clinics and hospital, security system, and health financing, human capital, and lastly is the health tech transformation. And it is categorized into two aspects or sectors. The first is the focus on information health technology. And the second one, the thing that we are going to discuss now, implementation of biotechnology in the medical field. So I would like to start with the introduction about the precision medicine, because today medicine is designed to target the average patient. So the approach is one fits all. It means that the possibility of the outcomes, it can be improved outcomes, or it can be no benefit, or it is even adverse side effect. Therefore, transitioning from conventional medicine into precision medicine is kind of necessity. So what I mean is by employing or implementing the newest technology in NGS and then sequencing analysis and other biotech technology, we can try to improve the healthcare, incorporating the genomic capacity in the hospital. Therefore, the treatment can be more personalized, more precise, and the outcomes would be improving result, and also reduction in the side effect. Why we are selecting the technology and why it is now? Because as you see, if we do it in year 20 years ago or maybe 25 years ago, it would be impossible due to the high cost. For the Human Genome Project, it was budgeted around 3 billion, but now look, the cost is declining and up to around 100. So it is the right moment. And actually, the pandemic era is kind of a blessing in disguise. Why? Because we have time to increase our capacity for sequencing, and the public also aware about the technology. Before pandemic, not many people, the public, accept the academia and research community knowing about the PCR, whole genome sequencing, the NGS, subvariant, and so on. But now it become public terminology. Therefore, we see the significant drop, making a affordable implementation in the large scale. And our minister, as you can see, the abbreviation is BGSI. It's similar with our Prime Minister name, Biomedical and Genome Science Initiative, will establish now, and we will try to cope with several actions. The first is establishing Indonesian Biobank, similar to UK Biobank. And also, at the moment, we are lacking information about genomic reference as a nation. We know that there is a reference genome GRC 38, and it was built from the Caucasian population. However, even the variation of genomes is still missing in Indonesia population. Therefore,